In the case of this paper, diversification or brain folds.
A neuroscientist started to look at the brain
computers, like the
more as an
NAFTA
this, but
interconnected
basically
system.
are very
incredibly negative
Area.
not what I
They're
described
powered
as
by
true.
all of you for being part of the largest attendance in the history of PETase. But anyway, here's Peter's final presentation. So yeah, round of applause for Peter, please.
All right. I'm going to be talking about plastic eating enzymes. This has been one of my interests pretty much since 10th grade. I apologize for my voice. Things happen. Okay.
Peter, is the plastic eating the enzymes? That's what you mean, right?
Definitely, yes. Okay, so I can start with why, because you should always do that. The papers I'm going to share 3 papers today. They're all of great importance to me. They kind of parallel my progression into research, into science, and I'll stop along the way and kind of explain the connections there. And then the other reason why I'm presenting on these papers is I think they're super exciting, they're inspiring, and they also leave me hopeful for the future. And we'll get into, unpack that more at the end, but I guess maybe you'll see. A little bit of, actually, there are no freshmen, right? Okay, anyways, a little bit of background. This is pretty much all the science you'll need to understand for the presentation. This is the central dogma. You start with your DNA in the nucleus, it gets transcribed into RNA, and then the RNA gets translated into proteins where you have like the codons that align with amino acids. Basically, the takeaway here is if you change the DNA, you can change the amino acids, and that will change properties of the protein that you're folding into. The other little bit that you'll need is we're focusing on a very specific type of protein. We're focusing on enzymes in this presentation, and you can basically think of it as a protein that will make a reaction, I guess, easier to occur, you could say, or requires less energy to actually start a reaction. And we'll talk about that a little bit more. Okay, so the problem. Plastic pollution is bad. Does anyone disagree? Raise your hand. Okay, Deshaun. All right, plastic pollution, not the best. It kills animals. It fills our seas. It looks bad. It's bad for the environment. The problem with plastic is that evolution hasn't really had the time to catch up, and so there's no way of kind of breaking down plastic, and that's why it stays around for so long. And then I guess our current solutions, right, you've all heard reduce, reuse, recycle. They're kind of insufficient to solving this problem, namely because there's already a lot of plastic already in the environment, and so reduce, reuse, recycle, I guess they're preventative solutions, which are great, right? But then there still is an issue where like we have a lot of plastic in the ocean, so how are you going to clean that up? So that's kind of the first problem. We already have plastic in the environment. The other thing is recycling is kind of cringe in that current methods require you need to heat the plastic up really, really hot, and that can cause deformations in the recycled bottles, so they're like less durable, which is problematic, and then it's also more like energy costly and thus price costly. So you kind of have problems with current recycling methods, especially for plastic. Okay, the plastic we're going to focus on is polyethylene terephthalate. It's the most commonly used plastic. It's a polymer, so it's basically this one segment of chemical that's repeated multiple times. So yeah, PET, that's the target. The first paper I'll be talking about is one of the first papers that I read. It's called A Bacterium That Degrades and Assimilates Polyethylene Terephthalate. When I read it, I was like, oh my God, this is the coolest thing. Yeah, I mean, it was kind of like a moment where I'm like, oh wow, we're like humanity's like not screwed. So a little bit of context. I was the kid in middle school that was sent to the equivalent of Aviva's office for going around saying, oh, we're all gonna die, it's not worth doing anything anyway. And some of that was climate change, but also that was just kind of like, yeah, life's useless, we're all gonna die, blah, blah, blah. But kind of, it's like papers like this where you're like, wow, like this is an enzyme, and I'll get into the paper more, an enzyme that can break down plastic, that can solve a big problem that current humanity is facing, that kind of has started to get me to change that mentality. We're all still gonna die, but like, might as well have a good life. Okay, so what this paper entails, a group of researchers in Japan went on a field trip to a recycling plant, and they walked around and they took a ton of samples. They sampled the soil around the plant, they sampled the plastic that was in the plant or being processed in the plant, and they took all those samples back to the lab, and then they just tried to grow any like microorganism off of those samples that they could. They grew a ton, and then I guess in all of the cultures, they put slips of PET. And so it was a plastic recycling plant, so they were kind of hoping, oh, maybe we'll find something that can naturally break down plastic. Turns out they did. So after waiting 70 days, which is a really long time, they eventually saw that in one of their samples, the plastic film was starting to degrade and starting to fray here. And so they took it out, looked at it under a microscope, and they did see kind of deformations in the plastic itself. And then also, as they were running these experiments, they were weighing the plastic to track its weight, and they watched the weight kind of go down. And so the implication here, right, is that there's some organism that's degrading the plastic. So they found that organism, and they sequenced that organism, and they found that there are two enzymes that are in the, I guess, chain of breaking down the plastic film, the PET film that they put in their cultures. The first one is PETase, which is the main thing I'll be talking about today, and the other one is enzymatic. To explain these two enzymes, I have a little activity. So this is PET. It's a polymer. It's made up of multiple repeating chemicals. PET is the enzyme that will cut each circle like that, all right? Okay? And then you have MHET, which takes the monomer form and makes it even smaller. Okay. And these smaller parts are actually okay for the environment. So that's like really, really cool. But we have another problem. PETase and enzymatic are really, really slow. If you go at the rate that it was going at in that little vial with the PET film, a typical plastic water bottle would take 34 years to degrade, which is not really a viable solution, and 34 is like kind of a back of the envelope like number, so like don't quote me on that. But the point is it would take a really long time, and that's not really a viable strategy if you want to use this to help degrade plastic that's already in the environment or to help with making recycling more efficient. And so we need to make it go faster. That's where the second paper of the story comes in. This is the first paper that I read. I was in 10th grade biology, and Tripp was like, here's a paper on kinetics, read it, and then next class we'll discuss it. I was one of two kids that actually read the paper. And I didn't understand one bit of it at all. But that was, thank you, Tripp. But that was completely okay. Tripp is a good teacher, right? So he sat us down and he kind of explained the paper. And then my first time presenting at Science Friday, I also actually presented on this paper, so I'm definitely not recycling slides or anything. But the point is, I was able to go from a paper of basically an alphabet soup of acronyms like PETase, enzymatic, blah, 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 all these assays. And with the help of great teachers at Nueva, and then also help of Science Friday, kind of shout out to them, I was able to get a fuller understanding or grasp on the paper. And so that was kind of the first time where I saw like progress in reading comprehension prior to like middle school where I took the same like ERBs every year and still got a bad grade on reading comprehension. So the point is, this was kind of where I started to see my scientific literacy increase, and so I think that was pretty cool. The paper mostly focuses on MHETase. It kind of gets like a crystal structure for it and then talks about the actual energy kinetics. We're not going to focus on that part of the paper. There's just one figure that I thought was pretty cool, and so we're going to talk about it. They kind of made this nice picture here, but they had one experiment in the paper where they would link PETase and MHETase together with these like protein chains. And so you have the two different enzymes, they're linked together, and they link them at 3 different lengths. And then they just tested to see how fast they were. So PETase and MHETase, perform kind of slowly when they're alone. They perform a little better when they're put together. But when they're linked together, they go a lot faster. And to be honest, I don't know why. You can guess like, oh, maybe it makes it easier to get like the substrate into MHETase, and then you get it all the way down or reduced or whatever, and you clear like the substrate so it doesn't clog up and have like unintended inhibition. But to be honest, like it matters less so why and more so that it actually happens. And so, and that's, I think to some extent, if you're in a class and you really just need a pass, you can just use that shortcut. I'm kidding. But the point here is linking the two enzymes together makes them a lot faster, right? And so now with that back of the envelope number, we've gone from about 34 years to maybe 6, 7 years, which is kind of crazy in terms of improvement by just like slapping a linker between the two enzymes. But we can go faster. 6 years for 1 water bottle, it's not fast enough. And that brings us to this paper, Machine Learning Aided Engineering for Hydrolysis for PET Depolymerization. So this paper, I'm going to talk about why I care about it, is the whole reason why I started an XRT project. Zach and I were in robotics, and we're talking about plastic-eating enzymes, as you typically would in robotics. And Zach was like, oh, I read this cool paper on how some researchers use machine learning to make PETase a lot faster. And basically from that, we're kind of like, this is a cool piece of software, let's kind of explore this together. We spent the whole next summer researching, and then we started our own XRT project, which has been a lot less successful than this paper, but a lot more educational than this paper. Anyway, what this paper does is the researchers developed a machine learning model. They trained it on a database of a whole bunch of proteins. So you basically got the sequence and then the folded protein in that database. And from there, you could get properties about what would make an enzyme more robust given a certain environment. From there, they could basically plug in the PETase gene, so that strand of DNA that I was talking about at the beginning with the central dogma, and the machine learning model would predict the specific amino acids along the protein that you could change, and it predicted what you should change them to. And those changes might make them more stable, might make PET more stable in different environments. So the researchers were kind of testing that model. They took the enzyme, they made the changes from the machine learning model, and then they tested the mutated enzyme to see if it would perform better or worse. I have no clue how the machine learning works. That's why Zach is also in our group. And then a shout out to Owen, who has helped a lot this year as well. All right, so on to, there are a lot of cool figures. There are only a couple that I'm gonna highlight. The first is this one. The mutant PETase that they developed, they called it fast PETase. And then the wild type is the one that we've been talking about all of this presentation. I think the graph kind of speaks for itself in terms of how insane this enzyme performs, but this is like a crazy improvement between the fast PETase and then the wild type. And then if you're wondering what these other lines are, they're different mutations from past papers. They're kind of bad, but fast PETase goes good. It goes hard. Okay. But like we care about actual results. So I've been talking about the plastic water bottle as my unit, and it was only for this slide. With this fast enzyme, they're able to degrade the plastic water bottle in about.
Hello, Nueva. At 12 o'clock, the rave will officially be open to general admission. IDs will be checked. Be there. Oh, student only.
All right, I'll try to finish before 12 then. All right, but yeah, 15 days for a water bottle. So kind of some scope, we went from 34 years to about 6 to 7 years to 15 days for one water bottle. That's kind of insane improvement. Cool. The other cool thing about being able to use this fast PETase enzyme is it kind of highlights potential for efficient plastic recycling. So current recycling, you need to heat up the plastic to maybe 200 to 300 degrees Celsius to recycle it properly. And that's really, really energy intense. In this paper, they're able to recycle this like plastic Tupperware at about 50 degrees Celsius, which is still warm, but that is a lot less energy than 200, 300 degrees. So there is potential for more efficient plastic recycling. I mean, also 15 days for one water bottle is still a bit slow, right? So we can still go faster, but I don't have another paper for you today. So one thing to be careful of, there's a lot of, the PETase is very, very common, right? And so this can potentially be used as a bioweapon if you wanted to like, I don't know, if you wanted to break down plastic that was essential for some structure or something. So I don't know, it's just a reminder that you should be responsible. Okay, but this progress leaves me optimistic, right? We started with this kind of this discovery phase where you have the researchers going and taking all these samples, and then you have tinkering with the original enzymes by just linking them with the protein chain, and then you can actually use it as a template to make this super enzyme, right? So like because of that process, we've gone from 34 years to 15 days in about 6 years, right? So big improvement, medium-ish time span, and then the implications, I guess, are twofold, right? So you can help with pollution, you can help with the plastic that's already in the ocean. So has anyone seen the sheeps or maybe the goats that are on the side of the hills like eating the grass? I imagine a world where instead of goats and grass, it's enzymes and plastic. I think that would be pretty cool. But the other application is more efficient recycling. So reduced temperatures are definitely going to be cool, especially in a day and age where we're getting more and more concerned about where the energy is coming from and how much energy people use. Okay, so this slide was kind of like shout out to research, but also kind of just like a thank you to research. The only reason why I'm standing up here today is because I've had the great teacher support, the great community support from JC and XRT to be able to understand these papers and kind of explore my interest in these realms. And so I'm very grateful that I've had that opportunity. I'm very grateful to the teachers that have helped me get to this state. And I encourage all of you who are interested in research, or maybe you're just interested in science, to maybe try XRTL1 or try JC for a month or something, and see where that takes you. Maybe just come to more Science Fridays. Like, I think it has made a net positive on my life, and so I would encourage and hope that it's the same for you. These are the 3 papers that I pulled up today, and of course we can go faster.